For the what time, sorry?
Clonotype?
Oh, clonotype.
Yeah.
Yeah, so it utilizes the file that comes with VDJ sequencing in order to identify the different genes associated with the different regions of the clonotype.
Yeah. I mean, so are you defining it based on like CDR3, or is it more of like a heavy chain, light chain pairing?
Yeah, it's a heavy chain, light chain pairing. We can also look at the CD3 here in the VDJ plugin. Let me show you. I think it'll become clearer once you open up that plugin.
Yeah.
So,
But I mean, yeah. I can wait, like, till we get there, so I'm not jumping the gun.
We're pretty much there, I think.
Okay.
Let's just see here. This is my demo data from 10x that we're currently looking at in the VDJ Explorer. If I go back to my desktop, I have your data there.
Yeah, we're really interested in that. Because you said you found something very neat.
Well, I just like the way that the plugin was currently analyzing the data. You guys would have to tell me whether or not what I found was very useful. And then the metadata is all stored in these different folders. So this all contain annotations. One of these files required a little bit of editing by us to make it fit the data, but I think we have it now to where it maps really nicely between the two.
Yeah. Now, one thing would be real good if you could send us what you found that works between the one and your concatenated.
I mean,
data.
it's just changing the dot versus dash, right? That was the problem.
Exactly.
Yeah.
Yeah. The cell IDs have to match.
Yeah.
And we
That's
did
an
send
Excel
back, I think, an example where we got that to work.
Which one is the concatenated?
Okay, yeah, I was just having that problem.
This problem.
Yep.
This is troubling. I wonder if I loaded the wrong annotations. The only thing I can think.
What about the bottom?
Yeah, I was just having that exact same problem.
Okay.
Yeah.
Well, it worries me for sure because I'm going to try just loading in the raw data and make sure I can connect before I created all these populations and kind of done this analysis. Ideally, I mean, it would work right here.
Yeah.
Let's just double check that everything seems right to begin with. Same idea here. Oh, I do remember. I think this edit is the one that has the correct.
That would be good to send that when it works. I always wonder if it doesn't work or it's me.
So there it worked. And make sure it works for 165 as well. But this has me worried because clearly we were trying to connect the exact same file a moment ago and it didn't work. Okay, so those two, these are the two pieces of meta information coming from the two concatenated files. And currently I can see that it's working because a huge number of the barcodes found in those two pieces of meta information were mapped.
Okay.
So this is what you want to see. Although ideally with some subpopulations with which to make comparisons.
Yeah.
Because without those subpopulations, it's a little bit meaningless.
Yeah.
So for example, let's see if I can manage to get a couple of subpopulations in there and map well. And specifically, might be interested in the two samples, right?
Yeah.
That didn't work. And I think ideally we'll be able to refresh. that, but with regard to those, I really like making this with those unbiased clusters. Plus, then you have a diverse range of populations.
Yeah.
Once you do get this mirrored, clicking Explore Repertoire will take us into this new kind of display for the data, and it gives this summary.
Yeah.
And so, as you mentioned, looking at the heavy and light chain combinations, we get a number of clonotypes.
Yeah.
And we can also segment this up by variable genes and diversity genes, and getting into some of the nitty-gritty details.
Yeah.
Now, what is a clonotype? What does that mean? Because there's more clonotypes than cells, but is that combined? 6,000 I notice.
Yeah, I go to the summary.
So 6,000.
I think I chose some bad metadata to load in here because it looks like all of the dashes in the gene names got replaced with dots as well. And that's why we're getting this clear distinction between the two samples.
Yeah.
Oh, there's definitely, back here, one of these data files has that corrected. I think it must be this file. You can see that, like, in the J gene, we have dashes, not dots. But in the barcode, we have dots. So that's what I did wrong there. I should have been loading this edit dots, or the CSV file that contains that edit string. You could probably continue on, but just so you know, that's a little bit, this is looking a little bit funny. Something else you can do here, filter on this information. So if you were interested in a particular variable gene, or a few particular variable genes, you can right-click on a header here. Take out my current filter, so nothing is currently selected. If I select 3 of these, and apply, it should now only be showing those that have matching filter criteria, those rows from the meta information. And now if I select them and click Create Population, it creates this new population, which it annotates with those genes I had selected. So if you had a very particular filter that you wanted to look at, this is kind of how you could get very, very fine-grained in terms of the cells you're looking at. Another cool trick here, let me try and remove this filter if I can. So that should be, we should be back to our total set of all rows. And if I just want the most frequent chronotypes, I should be able to right-click here. Oh, that broke. That might be, oh gosh. I should be able to shoot the number of top chronotypes that I wanted to generate. So I'll get populations based on the different heavy and light chain pairs. And it'll actually give me back, should give me back the amino acid sequences corresponding to each of those chains.
Quick question, Ian. So when you're looking at the top, I mean, even if you're just looking at the summary of all of them, right, you see that, or like just in the first page, when it's showing the counts for some of the clonotypes, I think the highest clonotype had just 3 counts in it, while some have 2, 2, 2, but the majority of them, for the defined clonotypes, only have 1 member.
Right.
I was expecting, that there would be, at least I was expecting that, for one particular heavy and light chain pairing and the different V, DJ, gene segment usages, that there would be at least more than 1 member in there.
Yeah, it seems like in a lot of cases there are very few chronotypes that repeat.
Yes.
And I totally agree. That was a little bit surprising for me, but I guess it just points to the, as long as it's getting correct results, it's pointing to the presence of a huge amount of heterogeneity among the chronotypes that have expanded up.
I think what it is, like one little base pair change or amino acid change, or not amino acid, base pair or then all of a sudden it says, okay, that's different, which it is different. I don't, is there a way to show what? Well, let me ask you one other thing. This is a little off, but how do we find the actual sequence for a particular V region, a VDJ? Is there a way? Is there a way to do that? I know there is.
If you say the particular sequence, you mean the amino acid sequence, or going back to the base pair?
Going back to the base pairs that came from the single cell.
We don't do that portion of it. You might be able to go back to like FASTQ files. That's where that's contained.
Yeah.
Right. And then how do we identify? So we want a particular one, or say 2 or 3, whatever. Anyway, we want a small number. If we go back, how do we find it in the FASTQ file? Do we have to put in a number or whatever for this?
That's a great question. You could export a subpopulation
Right.
from SeqKeep, and you could look at the cell ID, and I wonder if that would take you back to the
Yeah.
FAST file. But not being as familiar with that data, I'm not totally sure on that.
So how do you export? You export the cell ID. How do you do that? Let's say you have like one.
This would obviously export all of the data.
Oh, okay.
But when you do the export, the CSV file that's generated will contain those barcodes corresponding to the cell ID.
So we want to just. Can you show an example? Just like if you get one clone, just maybe just to make. At some point you'll have a few. Just so I can make sure I can do that, just like even just export one. Is that it?
No, this is one sample,
Okay.
and here's the second sample, and now let's try again.
Oh, you got a lot of cells. Okay. But anyway.
Yeah, so what I'm trying to do now is get down to like a single clonal type, and so I can show you how that export would look. Here we go. There's the all config annotation file for sample 166 and 165. And from here I can look at the mapping both of these samples. And before I do anything to break it, let's try the top monotypes again.
Okay.
Okay, and this is the kind of output that you'll get, which, as you were noting, mostly contain 2 or 1 cells.
Yeah.
So if you were interested, for whatever reason, in this particular cell,
Right.
pair of cells,
Yeah.
I can go right-click, export, and maybe I'm not interested in much else besides the sample ID.
Yeah.
Maybe I want to know what gene, a couple of genes.
Mm-hmm.
I could select this guy. Now I'm only going to export 2 genes for 2 cells. We'd expect to get like a 4x4.
Mm-hmm.
A 3x2. Yeah, 3x3. You'll have some annotations at the top as well as the cell ID. Okay. And I can open this into a new workspace, but we actually want to see. This is our CSV file, so opening that up in Excel. We didn't have any values for those 2 genes. But here's the cell IDs for those 2 cells.
Yeah.
I see. So that should give us back to the. Now the cell ID. Now, oh right, and then from that you get the heavy and light chain. Go back to that and in the database. And like that cell, the first cell barcode, then that go back to the FASTQ file and ask what is that heavy and light chain sequence. I don't know how to ask that, but
Yeah, I'm not sure either, and we don't deal with those FASTQ files yet.
Yeah.
It's interesting because I'm sure other people will be asking the same kind of question. And then obviously these are the amino acids associated with heavy and light chain.
Yeah. With the CDR3.
cells.
Ian, so when you're in with or when you're using the VDG Explorer plugin, is the only way, right? Because I think I sent you an email. So usually, right, when you're looking at, like, let's say the phenograph that has, clustered, maybe we have like 13 different clusters, but you're particularly interested in, let's say, like, you just want to look at two clusters that has been generated by the unbiased clustering algorithm.
Yep.
So you can compare the two clusters and look at, okay, what is the differentially expressed genes between these two clusters? Could you do the same type of analysis for the BCR data? Like, what is the differentially, expressed or used heavy chain and light chain pairings between these two clusters?
I think I would want to see a little bit more spelled out the workflow or the information you're trying to get out.
Yeah.
But the way you described it, I actually don't know that there's a way to do that.
Yeah.
If you had an idea, like we picked a couple examples here.
Mm-hmm.
If I wanted to know what upregulated two cells relative to the rest of the data,
Yeah.
we could figure that out. To do that, I would start with creating a Boolean not.
Yeah.
The reason you do that is to have something to compare against, right?
Yeah. Yeah.
Well, we keep comparing those two cells, which remind me again, it's this sample.
Mm-hmm.
No, that's the wrong one, sorry. This sample versus, or this population, two cells versus these cells. The other thing to keep in mind is when we do our p-value calculation for differential expression analysis,
Yeah.
we do a bond correction for multiple observations.
Yeah.
And here we'll only have two observations.
Mm-hmm.
So we need to change that, probably just turn it off. I think it's in the graphs section of the preferences.
Okay.
The p-value adjustment method can be changed to false discovery rate, bond harmony, or just turned off.
Okay.
Currently.
Mm-hmm.
So if I turn that off, and just open up the raw sample,
Mm-hmm.
I'm going to switch to gene view because we're asking about differential expression.
Yeah.
And then as a test, I'm going to use my not gate here,
Yeah.
and I'm going to compare it with, this guy.
Mm-hmm.
So there are some genes that are being, overexpressed in that clonotype relative to the rest of the data.
Yeah.
And now if I go into volcano plot, whoa, it looks funny.
It does.
I don't know if I've seen something quite like this.
No, this is,
I can already see that in terms of our non-adjusted p-value here, so just a regular manual EU test, there's nothing significant above this line in terms of upregulated genes.
Yeah.
There are genes that are upregulated, but not to a significant level.
Yeah.
So I could drop it down a little bit and just see what we get. Up in interest. And so these are the genes that are upregulated in that particular set of cells.
Mm-hmm.
Relatively, not significantly, but
Yeah.
I also wonder if we calculations it's a little bit funny there. Huh. And then certainly there's also downregulated genes here.
Yeah.
Mitochondrial genes.
Mm-hmm.
Oh, and here's the
yeah, that makes sense. And that's a gene itself.
But
the
Well,
other thing that
Ian, I had a question. I mean, this is jumping ahead of just a little bit, but when you sent your results to us, or to me, anyway, you had this pinwheel display.
Yeah.
Can you make one of those?
Yeah, that's just where I was going to go next.
Oh, okay.
And actually, this is another version of the plugin that we're hoping to release soon. It has a couple more options in this compare window than you'll see in the current release. One of which, this is very small table because we're just comparing between two populations. But this is actually a heat map confusion matrix showing the results from Sharon Jensen diversity index or information radius.